So an active transport moves molecule against its concentration gradient, so from the area of low concentration to the area of high concentration. So it supports this equilibrium. So here we have a primary active transport, secondary active transport, and vesicular active transport. So let's start off with primary active transport. So primary active transport uses the hydrolyzing of ATP to carry out the movement of molecule. So let's check out this example. So in this example, we have sodium, potassium, ATPase. So it's a sodium, potassium, ATPase. What it does, it takes 3 sodium from intracellular space. So from an intracellular space, it takes 3 sodiums and then release it to the extracellular space. And it takes 2 potassium from extracellular space and release it to intracellular space. So another thing to keep in mind is that the intracellular space has a lower concentration of sodium ion but a higher concentration of a potassium ion. In contrast to that, the extracellular space has higher concentration of sodium ion but a lower concentration of potassium ion. So therefore, the sodium-potassium ATPase are moving potassium and sodium against their concentration gradient. So let's see how sodium-potassium ATPase works. So here is a carrier protein. So it has a high affinity for sodium ion. So sodium in this diagram is the red circles. So they get attached to those carrier protein. And then ATP get hydrolyzed to ADP and energy, and it releases a phosphate group. So the phosphate group gets attached to the carrier protein, changing its conformation, and then it changes it further and release the sodium ion into the extracellular space against its concentration gradient. Since we know that we already have high concentration of sodium ion in extracellular space, now what it does, it takes 2 potassium ion from extracellular space, which is already lower in extracellular space, but we have high affinity of potassium ion on the carrier protein. So the potassium ion binds to the carrier protein, then it changes the conformation, phosphate group gets released, and then the carrier protein changes back to its normal configuration or its resting configuration. So by doing so, it releases 2 groups of sodium ion into the intracellular space. So that is how the primary active transporter works. It uses the energy from hydrolyzing ATP directly. Next we have secondary active transport. Secondary active transport uses the potential energy stored in the concentration gradient of one molecule to push another molecule against their concentration gradient. So let's suppose this one. So in this example, we are going to see how sodium glucose transporter work as a secondary active transporter. So here, this is the extracellular fluid, and this one is the intracellular fluid. So the intracellular fluid, as we know, has low concentration of sodium ion. However, there is a high concentration of glucose intracellularly. And in the extracellular space, we have high concentration of sodium ion, but low concentration of glucose ion. So how are we going to send glucose ion from extracellular space to the intracellular space? So we do that by using sodium glucose transporter. So what it does is that since sodium is always eager to go inside the cell because of the concentration gradient, because of the large concentration gradient, so the sodium glucose transporter binds sodium onto the carrier protein, and it binds glucose molecule on the other side. And since the sodium ion wants to go in the cell so badly that the carrier protein changes its conformation and releases sodium ion into the intracellular space, while also releasing the glucose ion into the intracellular space. So what kind of energy the secondary active transporter used? It used the potential energy stored by the concentration gradient of sodium molecules. Next we have endocytosis. Endocytosis is a vesicular active transport. So in endocytosis, the membrane indents and makes a vesicle and takes it in the cell. So for example, there's a line, this one is the membrane. So here we can talk about non-selective endocytosis. So non-selective endocytosis makes an indent and takes in a little bit of extracellular fluid and then makes a circle and makes it a vesicle. So this one is a non-selective endocytosis. While the receptor-mediated transporter, what it does is it exposes receptors on the membrane, and then the ligand bind. As it binds, it makes an indent in the vesicle, in the cell membrane, and with the receptor and ligand in the vesicle, it gets endocytosed into the cell. So that's how an endocytosis works. Next we have exocytosis. Exocytosis is just the opposite of endocytosis. It releases large lipophobic molecules out of the cell, and the vesicle from the cell membrane from the vesicle fuses with the cell membrane of the cell. Next we have phagocytosis. The phagocytosis forms a vesicle using a cytoskeleton. So the endocytosis makes an indent on the cell membrane, while the phagocytosis, what it does is use the cytoskeleton of the cell to make a vesicle. So it is to engulf large biomolecules, for example, bacterium. So the ATP is required to move cytoskeleton and for intracellular transportation of the vesicle. All of these vesicular transporter requires energy in form of ATP. I hope that helps. Thank you for watching.